Well, thank you for joining us for the second half of our innovation session today. I really appreciate you all joining us on this Friday afternoon or Friday morning. Getting a little ahead of myself. First, let me introduce our next panel of speakers. And our first speaker is Dr. Christine Lamonte. Dr. Lamonte is an assistant professor at the University of Washington in the Division of Nephrology. Her research focuses on integrating clinical epidemiologic and bioinformatics approaches to decipher mechanisms underlying diabetic kidney disease. Dr. Lamonte is also an alumni of AKF's Clinical Scientist in Nephrology program. And right before I turn to Dr. Lamonte, I remind everyone, please drop your questions in the chat box. We'll bring all of our speakers for this panel together at the end to answer those questions. But without further ado, Dr. Lamonte, I'll turn it over to you.
Thanks, Mike. Hi, everyone. Thank you for having me here today. My name's Dr. Lamonte, and today I am excited to be talking with you about the Kidney Precision Medicine Project and moving towards a molecular understanding of kidney disease. Next slide. So what is the Kidney Precision Medicine Project, or KPMP as we call it for short? The KPMP is a multi-year research collaboration of leading institutions from around the country that is designed to study patients with kidney disease. And the purpose of the KPMP is to better understand the mechanisms of common causes of acute kidney injury and chronic kidney disease. And at the core of the KPMP is really patient engagement. And what I mean by that is that we have patients sitting on all KPMP committees and working groups, including within the central hub, which is designing a lot of the aspects of the KPMP consortium. Next slide. So why is a kidney precision medicine project needed at all? And what is precision medicine? So precision medicine is the idea that treatments should be individualized to patients. That means that we should be able to identify the right treatment, the right intervention for the right patient at the exact right time. And patients want to know, when it comes to kidney disease, what exactly do I have? What will happen to me? What can I do about it? And what does it mean for my family? Patients want care that is specific to them. Meanwhile, historically, our treatment of kidney disease has been really a one-size-fits-all approach. Next slide. And what I mean by that is that historically, we haven't really been good at distinguishing between causes of kidney disease or treating those specific causes. Instead, what classically has been the case is that once people have kidney disease, they're given medications to slow the progression over time, but those medications aren't specific to their cause of kidney disease. They aren't treating the underlying problem itself. They're really just aiming towards slowing down the disease process to keep things from getting worse. And right now, acute kidney injury and chronic kidney disease are defined by measures like serum creatinine and urine tests, like how much urine you're making and urine protein excretion. But these measures are really not very specific at all, and they don't distinguish patients from one another. They just tell you whether disease is present or not. And the reason why we need a specific precision medicine project within people with kidney disease is because otherwise studies have been done in animal models don't really replicate what we see in human phenotypes, and can't really tell us the same things that are going on. And otherwise, there's also a very poor molecular understanding of all of the variety that we see in human AKI and chronic kidney disease. And because of that poor molecular understanding, we're not able to identify or prioritize targets for treating disease that would be effective. And unlike cancer, where a lot of research is based on identifying the specific or on taking the cancer tissue and identifying the specific molecular aspects of that cancer, a lot of research on kidney disease hasn't involved kidney biopsy tissue from humans. So we haven't really been able to look at what's going on in the kidney of the people that we're taking care of to tell exactly what's going on at the cellular molecular level and come up with treatments targeting that. Next slide. So I've talked about what the kidney precision medicine is about. Now, what exactly is the design of the Kidney Precision Medicine Project? So how exactly are we going about identifying these molecular subtypes of disease and getting to precision medicine for kidney disease? So what the Kidney Precision Medicine Project is doing is performing kidney biopsies, so getting kidney tissue from people that we normally wouldn't biopsy in the clinical world. These are people with diabetes, chronic kidney disease, hypertension and chronic kidney disease, and acute kidney injury. And the reason that we don't typically biopsy these populations is because just based on the clinical story, we tend to assume we know what's going on and give them that one size for all treatment that we have. So the goal of the Kidney Precision Medicine Project is really to take a deeper look into these common causes of kidney disease by getting kidney biopsies, biopsying these populations, and then applying to the kidney biopsy samples what we're referring to as molecular interrogation approaches, where we're looking at the specific proteins in the urine, we're looking at the specific proteins in the kidney tissue. We're looking at the genes transcribed in each cell in the kidney tissue, and we're linking these very specific molecular processes to clinical outcomes like ultimate decline in kidney function, etc. And the idea here is that we're going to take this molecular information to be able to tell people you don't just have diabetes and chronic kidney disease, but you have this specific type of kidney disease related to your diabetes. And this specific type of kidney disease related to diabetes requires this specific treatment. Next slide. The core values surrounding the KPMP are really centered on putting patients first. We see our patients and participants as the heroes of KPMP, and KPMP prioritizes participant safety in the biopsy process. We involve the patient and community in designing all aspects of the KPMP, and the other and in this information that we're generating, we really want to disseminate this information as widely as possible, so it can have the biggest impact. So all of this molecular data that's being generated from these kidney biopsies is being made openly available for researchers from around the country and around the world to use to answer important questions about kidney disease as a whole. And what that means is ensuring that the quality of the data that is being putting out is high, and that we are being very inclusive and collaborative with our colleagues. Next slide. Speaking with our patients and participants, and asking the question, why would folks be open to undergoing a kidney biopsy to answer questions about kidney health for research purposes? And the reason behind that is because people really care about kidney disease and want to know what's going on and want to make care better for themselves and other people with kidney disease. 72% of people on dialysis have kidney disease attributed to these common causes, hypertension and diabetes, that we don't fully understand at the molecular level. And chronic kidney disease disproportionately affects African American and Hispanic members of our population. Many of our patients carry genetic risks that we like the APOL1 or other gene variants that are associated with kidney disease that we have yet to do enough research on to understand how to target directly. And really, the care for most of our patients doesn't include getting a kidney biopsy and incorporating that into the decision-making process. And information is something that people ultimately want to have. And the goal here is that getting all of this new information about kidney disease will allow us to develop new and effective therapies that are really linked to what's going on at the biological level. And taking a step back, developing these treatments at the individual level that are targeting subtypes of disease will improve public health as a whole, just given how prevalent chronic kidney disease is in our population. Next slide. So I mentioned that patients are a key part of our planning within the KPMP. And we have a committee called the Community Engagement Committee, where we have a panel of patients that sit in all meetings and calls. We also have patients in our steering committee, so that is the leadership committee within the KPMP. And our patient partners were actually involved in the development of the informed consent form, so ensuring that the language coming from the consortium, explaining the idea behind the KPMP and explaining the risks and of undergoing the biopsy procedure, and the information that will come out of that is explained at the level that everyone can understand, and that our patients are truly informed when they're consenting to being part of this research. And our patient partners are also involved in adoption, in influencing KPMP policies and plans. So in addition to developing the informed consent form, our patient partners have been involved in developing forms that give results back to patients, and on developing a no fault harm coverage plan in case there are any complications after the kidney biopsy or other research procedures. Next slide. So I've talked about what the KPMP goals are, but what does the KPMP look like on the ground running? And this is a schematic of the structure of the KPMP. So at the center, we have the central hub and the KMAP project, the Kidney Mapping Atlas project. And the central hub is the core site that is involved in collecting all of the clinical and social data and samples, and providing quality control, project management, and administrative support for the entirety of the KPMP. Meanwhile, the KMAP, the Kidney Mapping Atlas project, is where all of this data is being stored. All of this molecular data about what's going on in the cells in the kidney biopsy samples that we're taking. All of this information is being de-identified and placed on an interactive kidney tissue atlas that can be accessed through the web and disseminated broadly to the research community. And on the outside are our recruitment sites, where our participants with acute kidney injury and chronic kidney disease are being recruited, and kidney biopsies are being performed. The tissue interrogation sites that are developing and using novel technologies to analyze this tissue at the molecular level, and the opportunity pool sites, which are sites that have applied for separate research grants to fill in the gaps of research within the KPMP. Next slide. And so this is, again, a schematic of the structure within the KPMP. And what I really wanted to highlight here is the bottom part of this figure, which is the work that's being done within the tissue interrogation sites. I keep mentioning that we're going to be assessing this tissue, on the kidney biopsy tissue, on the molecular level, but what does that look like? So what that looks like is applying novel imaging technologies that's using things like artificial intelligence and machine learning to read the biopsy images and take away from them features that the naked human eye may not be able to see. There's also acquisition of transcriptomic and proteomic data. So using certain technologies and probes, we can identify the genes that are being transcribed in each cell in the kidney, and the proteins that are in each cell in the kidney, and relate these to each other to understand the disease processes going on. There's also metabolomics, which is looking at the metabolic processes within these cells, which we link to the gene transcription and the proteins in these cells. So a lot of really exciting, cutting edge technology that's being applied to these kidney biopsies that in ways that we haven't really seen before to help us understand what's going on in these common types of disease. Next slide. So as of January of this year, the KPMP has enrolled 289 participants, 210 people with chronic kidney disease, 62 people with acute kidney injury. And we also have a population of 17 participants with diabetes who have not developed kidney disease that we're getting biopsies on to identify factors that could have been protective, to answer the question, why did these folks with diabetes for 50 plus years never develop kidney disease? What can we learn from them and take to our patients with kidney disease? Next slide. And I wanted to close with an example of what we've learned from the KPMP, what we can learn from the KPMP. This is from a clinical pathologic molecular correlation case that's been published in the Clinical Journal of the American Society of Nephrology back in 2022. And this is what it looks like to take a KPMP biopsy from the clinical to the molecular level, and what we can learn from that. And so this is a participant who is 66 years old, has type 2 diabetes, high blood pressure, hypertension, and had a paraprotein in their system. And there's ways that having that paraprotein in your system can cause kidney disease. This participant had type 2 diabetes for 5 years with no eye disease or neuropathy related to their diabetes, and they'd had hypertension for 30 years without complications like stroke or heart disease. They had some protein in their urine, and their serum creatinine was 0.87, which is typically in the normal range. And what the kidney biopsy showed on light microscopy, which are the images on the slide now, was disease features of disease related to hypertension, high blood pressure. On the images that we're seeing now, there was no indication that there was diabetic kidney disease, that diabetes was affecting the kidney. Next slide. But then when we applied our molecular technologies, so this is using transcriptomic data, so looking at the genes being expressed across different cell types in the kidney, we see enrichment of pathways involved in glucose metabolism and lipid metabolism. So enrichment of pathways that are suggesting that diabetes, while we're not seeing diabetes affecting the tissue itself on a structural level, we are seeing cellular changes related to diabetes. So even though looking at the biopsy, we could not say there was diabetic kidney disease going on, looking at the molecular data, we can say there's the beginnings of diabetic kidney disease going on. So maybe the treatment for this patient shouldn't involve just hypertension, hypertensive kidney disease, but should also treat diabetic kidney disease. Next slide. And this is what, a global summary picture of what this molecular data looks like when you map it across different cell types in the kidney. As you can see, there's a lot of data, and the goal of the KPMP is to figure out ways to reduce this molecular data to disease pathways that can be actionable. Next slide. And so where will the KPMP lead? We are hoping to enhance molecular understanding of known clinical phenotypes, diabetic and hypertensive kidney disease, new mechanism-based disease subtypes, so saying you have one specific type of diabetic kidney disease versus another and treating those differently, biomarkers in the blood and urine to be able to diagnose those subtypes without a kidney biopsy, and also precision clinical trials. So once we identify those specific subtypes of disease, designing research studies specific to those groups to come up with therapies for those specific groups. And we're hoping that these aspects of precision medicine can be layered on population health. So broadly treating things like hypertension and diabetes in the community to overall improve health. Next slide. And this is my last slide. I just wanted to share the KPMP, a subset of the KPMP publications today in case folks want to learn more about the consortium and learn about the molecular data that's coming from this and what we've learned so far. Next slide. And I just want to thank all of my mentors and research collaborators at the University of Washington, the American Kidney Fund for supporting my research and having me here today, and the NIH. And thank you.
Thank you so much, Dr. Lamonte, for your work on the KPMP and sharing it with us today. It's clear that your research is really going to help to lead to more personalized treatments for people with CKD and acute kidney injury. If you're just joining us, please drop your questions into the chat box on the side. We are going to bring back all 4 of our speakers at the end for a few questions, so we'll have time for that. Our next panelist is here to discuss a drug that can impact people who have experienced acute kidney injury, or AKI, specifically during heart surgery. I want to thank Alexion AstraZeneca Rare Disease for sponsoring today's discussion on the Artemis study. And now I'd like to introduce Dr. Pamela Winterberg. Dr. Winterberg is a practicing pediatric nephrologist and researcher for 10 years before joining Alexion AstraZeneca Rare Disease in 2022. As Senior Medical Director in Clinical Development, she is dedicated to finding new treatment options for people living with kidney disease by designing and running clinical trials. Dr. Winterberg will be sharing with us information on the Artemis study. Dr. Winterberg?
Thank you, Mike. There we go. So yes, I am pleased to share with you the information about the Artemis study, which is aiming to reduce acute kidney injury for patients living with chronic kidney disease who need heart surgery. Next slide. So what is acute kidney injury? AKI is a sudden decrease in kidney function that can be seen hours to days after damage that occurs to the kidney cells. Signs and symptoms can include changes in the blood creatinine level, which is a marker of kidney function, as was previously mentioned, decreased amount and frequency of urine, changes in the blood acid and salt levels, which are normally balanced by the kidneys when they're functioning well, or buildup of fluid in the body. That can be seen as swelling, fluid in or around the lungs, or fluid around the heart. Next slide. So in general, the causes of AKI are when kidney cells can be injured by either decreased blood flow to the kidneys, or in some instances, certain medications that can damage the kidney cells. The Artemis study is primarily focused on the decreased blood flow to the kidneys during heart surgery. Next slide. Unfortunately, there is currently no approved treatments that specifically prevent or treat AKI. So instead, AKI is managed by using medicines to increase the amount of urine when appropriate, restricting certain types of nutrition or medications that can build up in the body during AKI when the kidneys are not functioning fully, using temporary dialysis to clear the blood and remove excess fluid, and supportive care in the intensive care unit while waiting for the kidneys to heal and recover more function. Next slide. So why are we studying AKI during heart surgery? There's a few reasons. First, certain types of heart surgery use a machine called the cardiopulmonary bypass, or CPB. And in those types of surgeries, there are high rates of AKI. So there's a large unmet need there. Next, it can take several days after the injury has actually happened in order for us to detect the AKI using creatinine. And by then, the injury may be so widespread, it's hard to treat. So rather, we're looking to prevent the kidney injury because it has a higher likelihood to decrease the complications of AKI compared to treating AKI after it's detected. Next slide. Kidney injury is a known complication, especially in patients with CKD who are undergoing heart surgery. And assuming that the kidney injury event itself is occurring during or shortly after the surgery, identifying the patients that are at the highest risk of this injury provides an opportunity to consider a treatment before the surgery to reduce the kidney injury events during and after surgery. Next slide. So a little bit more about what CPB is. It is a machine that temporarily takes over the function of the heart and lungs during the surgery on the heart. So this machine adds oxygen to the blood, and then it pumps blood around the body. And this allows the surgeon to perform the procedures on the open chest in the heart. Next slide. Unfortunately, the kidneys and kidney damage and inflammation create a vicious cycle during cardiopulmonary bypass. So first, the changes in the way the blood is flowing through the body during CPB can injure kidneys. CKD kidneys may actually be more sensitive to this type of injury. Second, once the kidneys have cell damage, they can trigger inflammation. Third, inflammation, either in the blood vessels or released by the kidney, can then feed back and further injure the kidney. So there's a vicious cycle between the kidney damage and inflammation. Finally, once the patient is taken off of the CPB pump and normal blood flow is returned to the kidney, it can actually shock the kidney cells, leading to more damage. So in summary, inflammation in this vicious cycle can be started or worsened by uncontrolled activation of a part of the immune system called complement. And it's this complement system that is the focus of the Artemis study. Next slide. So how do we find new treatments for AKI? In general, a clinical trial is needed, which is a scientific study designed to answer the following questions. Is a treatment effective, or does it work? And is a treatment safe? Next. So the Artemis study is a phase 3 randomized, double-blind, placebo-controlled clinical trial to study whether blocking complement activity reduces the risk of kidney events in people living with chronic kidney disease who need heart surgery. Next slide. There were a lot of vocabulary words in that statement, so let's break that down a little bit. Phase 3 is the terminology used to describe the ultimate study that's required for approval by the Food and Drug Administration, or FDA, for a medicine to be sold as a treatment for a specific disease or condition. These types of studies often include information from several hundred to thousands of people about the safety and efficacy of the treatment under study. Placebo-controlled means that some participants in the trial receive active medicine, while some participants receive an inactive substance that looks exactly the same and are indistinguishable. Randomized means that each participant's type of treatment, either the active medicine or placebo, is chosen at random during the trial. And finally, double-blind means that the study participants, the healthcare providers participating in the trial, and the sponsor of the study do not know which individuals in the study receive the active medicine versus the placebo until all the participants have finished the study and the data is analyzed. Next slide. So here's a brief overview of the Artemis study. The study is planned to include 736 participants, half of which will receive a single IV dose of placebo, and the other half will receive a single IV dose of ravulizumab 1 to 7 days before a planned heart surgery with cardiopulmonary bypass. There's a 28-day screening window prior to the dosing in order for participants to sign informed consent and to have a screening visit where the study team at the local hospital will determine a participant's eligibility for the study. After surgery, there are several study visits through 90 days after the surgery, at which time we'll be looking at what are called major adverse kidney events, or MAKE. So at the end of the study, we will be comparing the number or percentage of participants receiving placebo versus ravulizumab who've experienced MAKE at 90 days. So let's see what that means on the next slide. So New treatments for AKI are tested currently based on reducing the events after the AKI or the injury itself. So this is where what they call major adverse kidney events, or MAKE, in the orange box. So a participant is identified as experiencing a MAKE event if they have either died within the 90 days, have needed new dialysis within those 90 days, or if their kidney function is decreased at 90 days. And for this study, it's defined as at least 25% lower than the kidney function they had at the prior to the surgery. Next slide. Clinical trials are determine who is eligible by both what are called inclusion criteria and exclusion criteria. So inclusion criteria is the first step to determine who's eligible. The Artemis study is enrolling adults aged 18 through 90 years who are needing non-emergency heart surgery for conditions such as coronary artery disease or heart attack, or heart valve diseases. Participants need to have chronic kidney disease with a current GFR, which is a measure of kidney function, between 20 and 60 mils per minute. In addition, the individual participants need to have a certain number of risk factors that also highlight the increased risk for kidney events after surgery, and that's determined by the study team at the hospital. Next slide. There are a few exclusion criteria which identify participants who may not be eligible. Those include anyone who needs emergency heart surgery, or if the surgery is planned without the use of the cardiopulmonary bypass machine, called off-pump surgeries. Additionally, if the participant is already receiving dialysis before the heart surgery or has experienced an AKI event in the 30 days prior to surgery. And then finally, participants or patients who have received an organ transplant before surgery, including bone marrow transplant, are excluded. Next slide. So in summary, AKI during and after heart surgery can be caused by changes in the blood flow to the kidneys and inflammation that occurs during the cardiopulmonary bypass pump part of the procedure. People living with CKD are higher risk for AKI and those potentially the major adverse kidney events after heart surgery. The Artemis study is evaluating the impact of a new treatment aimed at blocking the complement and inflammation during surgery on those kidney events after heart surgery in high-risk patients who have chronic kidney disease. Next slide. For additional information, you can visit the Artemis study website listed on the slide. In addition, the trial, which is a global trial, is also listed on clinicaltrials.gov. Thank you for having me and letting me describe the study.
Yeah. You're very welcome, Dr. Winterberg. Thank you for doing it. And my team, I think, is dropping in a link to the chat as well. So there's a clickable link. You can learn more about the trial right there in the chat box. And the chat box is a great place to also drop your questions. I'll remind everyone again to do that. We're going to bring all of our panelists back in at the end to answer your questions. Keeping right along with talking about some trials that are happening, I want to bring in our next panelist, Lauren Eva, and she's here to share about NephCure's work with the Parasol Project and what they're doing to help design treatments for focal segmental glomerulosclerosis. You've probably heard of that as FSGS. But now I'd like to welcome in Lauren. Lauren is NephCure's Executive Vice President of Professional Relations. She relishes every opportunity she gets to spend with NephCure families and is constantly learning more about the patient journey. Since joining NephCure in 2014, she's thrilled with the recent pace of new discoveries in genetic testing, clinical care, and potential new treatments for patients and families affected by rare protein-spilling kidney diseases. Lauren?
Thanks, Mike, and thanks for having me here today. I'm really excited to tell you about this new project that really recently launched. It's called the Parasol Project, and it has to do with facilitating drug development for a rare kidney disease called FSGS. So I'm going to tell you a little bit about this and how the larger kidney disease research community is coming together in a really impressive and unprecedented way to support and speed new treatments for this condition. Next slide, please. So just quickly, NephCure is a patient advocacy group. We are, our mission is to empower people with rare protein spilling kidney disease to take charge of their health while leading the revolution in research, new treatments, and care. And so we're a nonprofit that pushes rare kidney disease research and care forward while connecting patients, families, and caregivers to the resources that they need. Next slide, please. And so we really just serve primary rare protein spilling individuals with primary rare protein spilling kidney diseases, including those you see listed here. Next slide, please. And so to understand why we need new FDA-approved treatments for focal segmental glomerulosclerosis, or FSGS, let's first just talk a little bit about this condition. So FSGS is a rare disease. It's characterized by scarring of the kidney's filtering units, and it impacts about 40 to 60,000 people in the United States. This is a primary disease. It's not caused by other conditions like diabetes or obesity. And there are different subtypes. It can be autoimmune in nature or genetic or undetermined. FSGS is progressive and aggressive. Up to 50% of people will progress to kidney failure. And it looks different in each individual. We often say when you've met one person with FSGS, then you've met one person with FSGS. And so there's not really a uniform prognosis, and it's difficult to predict. I'll talk a little bit more about that later. Currently, it's treated initially with prednisone, and if that does not have an impact on putting the disease into remission, then other immunosuppressants are used, often with increasing degrees of toxicity or intensity. These can sometimes be drugs that are also used for chemotherapy. And so the side effects of these drugs are often difficult to deal with and quite intense. Some people with FSGS respond to treatment and go into remission. Some people never respond to treatment and progress to kidney failure. Some people respond, but then relapse when they're taken off of their treatments. And so, again, like I mentioned, it's difficult to predict, and there's a lot of different journeys. For individuals that have genetic causes of FSGS, they are typically unlikely to respond to immunosuppression. And so there's currently no FDA-approved treatments. All of the treatments that are currently used are off-label. And who is impacted by FSGS? It is a disease that impacts people in the Black and African American community 4 to 5 times more often. And it's more common in adults, but it is also the leading single disease cause of kidney failure in children. Next slide, please. So just quickly, a quick primer on the FDA drug approval process. So to be approved by the FDA, a drug must demonstrate substantial evidence of effectiveness and clinical benefit. And so clinical benefit is really the impact of how a patient feels, functions, or survives on treatment. And that we see an improvement or delay in the progression of clinically meaningful aspects of the disease. Next slide, please. And so how do we measure the effectiveness of drugs? So we use a thing called endpoints, a clinical trial endpoint. This is an event or an outcome that can be measured objectively to determine whether the intervention that's being studied is beneficial. And so some endpoints that might be, easy to imagine or to understand these would be like overall survival, if someone, survives on a drug or does not. If there's events like stroke, heart failure, et cetera, hospitalizations or reaching end stage disease. And so you can also think of an endpoint sort of like a goal, although we're really just using it to understand the performance of the treatment. Whether or not the drug being measured or the treatment being measured reaches the endpoint or the goal. Of course, we want new treatments. We want things to reach their primary outcome and their goal, but it's all data that helps us better understand the disease or the pathway or what drugs to study next. And so just having the clinical trial, being able to measure the drug and get the results is the win. Next slide, please. So for FSGS, there's drug approval processes. There's many different drug approval processes, but relevant to FSGS is really 2 pathways. one called the traditional approval pathway and then the accelerated approval pathway. And so the traditional approval pathway is used for all patient populations. This is what you might be more familiar with. This slide shows a comparison between the traditional and the accelerated approval pathways. And so you'll see that in both, there are rigorous high standards for the study design and the data collection and data interpretation. In a traditional approval pathway, a primary endpoint is used to show the clinical benefit. So like some of those endpoints, some of those outcomes that I just covered, end stage disease, heart failure, hospitalizations, mortality, those are primary endpoints. The review time is typically 1 to 2 years, and the study sponsor or the pharmaceutical company would be required to conduct surveillance of the adverse effects of the treatment once it's been approved. But in an accelerated approval pathway, this is a relatively new approval pathway that's offered for diseases that have a high unmet need. And so the disease might be particularly aggressive, or it might be considered an orphan disease or rare disease. And so in the accelerated approval pathway, rigorous high standards for study design and data collection are also used. But what's different is that there's a surrogate endpoint that can predict clinical benefit. And so a surrogate endpoint is used when the measure of the clinical outcome might take a really long time to study and might be what's called not really feasible necessarily to study in the confines of a clinical trial. So it might take too long, be too expensive and costly, and ultimately deter biopharmaceutical industry from really investing in discovering new treatments for this disease. And it's also used when the clinical benefit of proving the surrogate endpoint is well understood. And so what's different about the accelerated pathway, as well as, you see in the name, is that the review time is much shorter. So there's a 10-month review time. And so what that means, if you go to the next slide, is that the drug is able to get approved and be on the market and be prescribed and in use for patients faster than in the traditional drug development pathway. But the study sponsor runs trials after the approval, confirmatory trials, to validate the effectiveness of the drug. So there's post-market confirmatory trials. And so next slide, please. And so for kidney disease, the clinical trial endpoints that are available to use. So really the FDA wants to know if a treatment will help kidneys survive. And so glomerular filtration rate, the decline of glomerular filtration rate, is already used as a surrogate endpoint for kidney failure or a substitute endpoint for kidney failure. And this is a way to measure or predict if kidneys will fail in a shorter time frame. And so the FDA currently accepts the halving of glomerular filtration rate as a surrogate endpoint for the development of kidney failure in clinical trials of kidney disease progression. And something that is relatively new is the use of proteinuria, or the measurement of protein in the urine, as a surrogate endpoint for GFR decline. And proteinuria was accepted as a surrogate endpoint for IgA nephropathy in 2019. There's a paper that was released. I put a screenshot of this. It was published in CJSON. And so once that was accepted, proteinuria reduction is accepted now as a substitute, or surrogate endpoint in clinical trials for IgA nephropathy. We saw a lot of investment from the biopharmaceutical industry in trials and research for IgA nephropathy, which is wonderful. And what that led to was the accelerated approval for some new treatments, some first ever FDA approved treatments for IgA nephropathy in 2021 and 2023. And then just this year, one of those treatments that was approved on the accelerated approval pathway process was granted full approval in January of this year. So that's really exciting. And that is, a potential model for how we can approve more drugs for kidney disease and more drugs for rare kidney diseases. So next slide, please. And so what's really unique and different about FSGS, especially in comparison to IgA nephropathy, but other rare kidney diseases as well, is that this is a disease that is difficult to predict and study. There's unique challenges of FSGS. This slide shows a charting of different trajectories of patients with FSGS pulled from an observational study called Neptune that shows that, this is a disease with loss of kidney function over years, so it's progressive. But there's what's called relapsing proteinuria. And so, as I mentioned before, patients will cycle between remission and relapse. There's not a straight trajectory. You can see that different patients have different experiences with their disease. There's also, this is a disease, like many primary kidney diseases, it's named by what pathologists see when they read a kidney biopsy. And so it's a grouping of several subdiseases, as I mentioned. And so there's some underlying differences or heterogeneity in terms of what causes FSGS. And so there's many different journeys that makes it really hard to necessarily see a strong signal from a treatment's effect. And so there've been many studies that have supported the use of protein in the urine, proteinuria, as a potential surrogate endpoint for FSGS as well. But we don't currently know the optimal model for the use of proteinuria as a surrogate endpoint in clinical trials for FSGS. So next slide, please. And so thus was born the Parasol project. And Parasol is a project that was created less than a year ago, really, to pull together existing data that exists about FSGS patients from both observational studies as well as drug studies, so that we can pull everything together and develop a new model that could describe the relationship between a surrogate endpoint and the clinical outcome that we could use to verify the clinical benefit of a drug after accelerated approval. And there's current trials with FSGS. There's been some trials in the past with FSGS as well. And the goal, as well for NephCure, because FSGS, this is a patient population that we serve, we're highly interested in supporting these ongoing and future trials to gain new options for treatments and to understand the approval pathways for FSGS. And so we need to better understand how to measure protein in the urine and GFR as predictors of kidney failure and disease progression in FSGS. Next slide, please. And so, as I said, this is really an unprecedented effort in FSGS. This has never been done before. There's coordination from all over the globe of basically every set of data, every observational study or drug study that has data on FSGS patients. They're all coming together. Everyone has agreed to coordinate together and pull this data. And what's happening now is that there are biostatisticians from several academic centers, as well as regulatory agencies that are all working together to harmonize this data so that we can use it to better understand how to predict, like I said, the impact of some of these potential surrogate endpoints on kidney failure and kidney decline. So currently there's 21 registries or data sets with FSGS patients that have come together. And we are working now on developing this model, describing this relationship between a reasonably likely surrogate endpoint and the clinical outcome. What will happen next is that we will create a suggested model. It'll be reviewed by all of the stakeholders, including our regulatory partners, which means the FDA will be able to review this model, as well as the European Medicines Association. And the goal is really to have a published paper with the results, that the results are ready by the end of the year. And so this is a project that would typically take 2 or 3 or more years to complete from start to finish. As I mentioned, we launched this project less than a year ago, and we're hoping to have the results before the end of the year, by October or November of this year. And so in the next slide, you can see that this is a joint effort between many organizations, NephCure, as well as the International Society of Glomerular Disease, which is a physician organization, in partnership with the Kidney Health Initiative, which is a public-private partnership between ASN and FDA, as well as the National Kidney Foundation, have all come together to organize and manage this project. The University of Michigan is managing the harmonization and the interpretation of the data sets. And there are many other individuals and organizations that have also contributed to this massive project. And so we are excited to have the results before the end of the year.
Great. Well, thank you very much, Lauren. And my team is putting some links in for how you can learn more about the Parasol project that's moving forward. We always appreciate the work in partnering with NephCure on this. They're making a difference for sure for patients. Our next panelist is here to share about their research that is impacting a specific group of patients with kidney disease, those with APOL1 gene mutations. I'd like to introduce Dr. Annika Lucas. Dr. Lucas is a past ACAF's clinical scientist and nephrology fellow from 2020 to 2022 and is currently a nephrologist at Duke University. As a clinician investigator, her aim is to improve the quality of life and health outcomes of Black patients with kidney disease. Dr. Lucas, could you please share with us the work you're doing on the Justice study?
Thank you so much, Mike. Hello, everyone, and thank you to the AKF for having me. As Mike shared, I'm a nephrologist at Duke, and we'll be discussing the Justice trial. Next slide, please. So here's an outline of our talk today. First, I'm going to review APOL1 mediated kidney disease. Then I'm going to describe the goals of the Justice trial and also review eligibility criteria for the study and the study components. Next slide, please. So there's a disproportionate burden of kidney disease among Black Americans. We know that Black Americans are 4 times more likely to develop kidney failure compared to white Americans. And there are many factors that contribute to this racial disparity, including social factors, things like limited access to healthy food, poverty, environmental toxins. But for the purpose of the talk today, we will focus on biological factors which may contribute to this kidney disease. Next slide, please. So we know that among patients in general, there's a lack of kidney disease awareness, but especially in the Black community. 9 in 10 people with kidney disease do not know that they even have it. And here you can see a picture of many Black celebrities who actually have kidney disease. Next slide, please. So we know that there may be a biological basis for this racial disparity in kidney disease, as I described before, and that patients of West African ancestry may carry mutations or alterations in the apolipoprotein L1 or APOL1 gene. And these changes in the gene were protective against a parasite infection in sub-Saharan Africa called trypanomyosis. And patients who have 2 copies of these altered genes may be at more risk for development of kidney disease and kidney failure. And this mutation, as you can see, accompanied individuals of West African ancestry from the continent of Africa and traveled to the Americas. Next slide, please. So we know that approximately 4 in 10 Black Americans on dialysis have kidney failure caused by APOL1. So Black Americans represent about 10 to 15% of the population, but we know that in many dialysis units can represent about 40% of patients in kidney failure. Next slide. So why this study? So in Justice, we represent a group of kidney doctors, community leaders, and lay people who want to see a change. We want to reduce the burden of kidney disease among Black Americans. This study is funded by the National Institute of Health, and we know that about 40% of Black patients with hypertension associated end-stage kidney disease have this high-risk APOL1 genotype. And about 70% of Black patients with idiopathic FSGS have, again, this high-risk APOL1 genotype. So we know that there are 4 issues that we're dealing with here. This, as I mentioned before, this high burden of kidney disease and kidney failure in the Black community, lack of kidney disease awareness, and also lack of a specific treatment for the type of kidney disease. There's no approved treatment currently for APOL1 mediated kidney disease, but we do know it's the most common biological driver of this racial disparity in kidney health in the United States. And so with the Justice Trial, we hope to raise awareness through kidney disease education and also screening, but also provide options for treatment for APOL1 mediated kidney disease. Next slide, please. We know that Black Americans are underrepresented in clinical trials in kidney disease. When you look at many of our major studies, less than 5% of study participants were Black. And so with the Justice Trial, we want to address these obstacles and encourage participation of Black Americans into clinical trials. Next slide, please. So that is the name Justice derives from the treatment that we're offering, which is Janus kinase STAT inhibition to reduce APOL1 associated kidney disease. So we know that high expression of APOL1 causes kidney injury. The JAK-STAT pathway pictured here drives the expression of pathogenic APOL1 in kidney cells. And we know that preclinical data with baricitinib, which is a medication that is has been previously approved for the use of other diseases, such as rheumatoid arthritis, and has been proven to be safe in humans, has been shown to block this pathway and treat APOL1 associated kidney disease in preclinical data. And so with this, we hope that this may be an effective treatment. Can you go to the next slide, please? And so the Justice Trial is a phase 2 double-blinded randomized placebo-controlled trial using baricitinib. So as a phase 2 trial, there are 2 main goals. One is to see how well is this treatment, and then the second thing is what is the safety profile of this medication. So we want to evaluate the efficacy of baricitinib to reduce protein in the urine in Black Americans who have APOL1 associated FSGS and also who have hypertension as the cause of their kidney disease. And again, we're also going to be testing or monitoring the safety of this medication. Next slide, please. So in terms of our study overview, we have another study that I just want to allude to briefly called CARE. And with this, it's called the Community APOL1 Research Engagement, where we're going out in the community and we're genotyping individuals so that they know their status, to know if they are carrying this, this gene, this high-risk gene. And we're also checking people, testing them for the presence of kidney disease. And so with the CARE work that we're doing, not only in our local community of North Carolina, we are able to do this nationally. And I will share information about how you can get information on getting tested yourself. We are looking to see if patients have high amounts of protein, their GFR, which is a measure of kidney function, is greater than 25 or 26, and they're carrying the high-risk APOL1 genotype, then we see if they will be able to join the Justice study. And once you're enrolled in the Justice study, individuals have placed in either the placebo arm or the active drug baricitinib. So it's a double-blind trial, so you're not going to know that which form of the drug you're taking, if you're on placebo or if you're on active drug, until the conclusion of the study. And the study lasts about 6 months. Study participants will have to initially come to North Carolina, and we provide free transportation and accommodations for the initial study visit for the enrollment, and do laboratory testing, which is blood work, and then also urine sample as well, checking for the protein levels in the urine. And after that initial enrollment visit, individuals can have lab testing done remotely wherever you are in the United States once a month, and we will be able to get the results of that. Next slide, please. Thank you. So the key study outcomes. So we're interested in looking at the percent change in urine protein from baseline over the study period of 6 months, as I said before. And the secondary outcome, we're looking at percent change in the estimated glomerular filtration rate, or the measure of kidney function, over the study period. And finally, we're going to be monitoring for side effects for this medication. Next slide, please. So who's eligible to participate? So individuals who self-report or identify as being Black or African American, Afro-Caribbean, Afro-Latino, or anyone with recent African ancestry is eligible. You have to be between the ages of 18 to 70 years of age and have the high-risk APOL1 genotype. If you don't know your genotype, it's okay. You can reach out to us. We're happy to provide you with information about what your genotype is, all free. And individuals, again, who also have a history of hypertension or high blood pressure as the cause of their kidney disease, or have a diagnosis of FSGS that was proven by biopsy as well. You'll need to have about 300 milligrams per gram of protein in the urine and have an eGFR greater than 25 to 26 and be able to provide consent. Next slide, please. So we do have an extensive exclusion criteria, but some of the highlights: you cannot have a kidney transplant, have a history of diabetes, HIV, or sickle cell disease. In terms of immunosuppression, there are certain medications that if you're on things like cyclosporin, rituximab, that you will not be eligible. If you have any questions about that, feel free to reach out to us. Next slide, please. So we are happy to partner with different organizations in the community. We've partnered with the Seventh-day Adventist churches, Baptist Convention, AME Zion churches as well, and other nonprofits. We are happy to partner with other organizations as well. You can find more information about the Justice Trial at www.kidneycareandjustice.com. I just want to recognize our PI, Dr. Obayemi Olabisi, along with our co-investigators and our community partners listed here. Thank you so much for your time.
Well, thank you very much, Dr. Lucas. Your work in this area is so important to improve outcomes for people with APOL1-mediated kidney disease. As part of our work to increase awareness for AMKD, we're launching the first ever AMKD Awareness Day on April 30th, 2024. I'll ask my team to drop some information in there. But that's an opportunity, for communities, organizations, and families to better understand AMKD and become more APOL1 aware. So now I'd like to bring all of our panelists back in. We've got time for just a few questions, probably maybe one for each panelist. And Dr. Lamonte, I will come to you with the first question. Speaking, it just made me think of that and talk about AMKD. Are you looking at that with the KPMP and looking at genetic factors as well as you're doing the biopsies, or any future considerations to doing that?
The KPMP is performing genome sequencing, so we'll be collecting genetic data at the genome-wide level as well, in addition to transcriptomic data at the kidney tissue level. So yes, that is a data point that will be collected by the KPMP. Excellent.
And just a reminder, if there's any other questions coming in, please feel free to do those. Send us the chat and we'll get those over. Dr. Winterberg, I'll come to the next question for you. What sites are participating in Artemis, and can people across the country participate if they meet the requirements?
So we are, the clinical trial sites are actually in 19 different countries around the world, including the US. Probably the best place to see the list of the trial sites in the US is either the clinicaltrials.gov website or the Artemis study website. In terms of participating in the trial, potential participants need to be actually receiving their heart surgery at a trial site in order to participate. So it's open to anybody who's available to have their surgeries at those sites.
Okay, excellent. Lauren, let me come to you with a question. You know, you've done work in IgA nephropathy and now FSGS. What's next for Parasol coming up?
Yeah, once we reach our goal at the end of the year and release a new model for FSGS, we're really hopeful that this effort that has brought together these partners from across the world can continue to look at other rare kidney diseases and identify more models for diseases like membranous nephropathy or C3 glomerulopathy, or even some of the sub diseases of FSGS, like APOL1 kidney disease, and additionally recurrent FSGS after transplant.
Great. Dr. Lucas, a question for you. How often will patients need to visit the study site to participate in Justice, and what procedure is done during these visits?
Great question. So participants only need to come to the study site once. That's that for enrollment, and then subsequently they can do blood testing and urine testing remotely wherever they are, and we will receive the results of that. And so it's very accommodating to participants. And really what it entails is really blood and urine testing for subsequent visits that can be, as I said, conducted remotely.
Great. Lauren, one more for you. One second. You know, this is a question actually related to some of the sessions we've had earlier in the week, but how often is FSGS undiagnosed, misdiagnosed, and what can we do about it to kind of help with that?
Yeah, great question. And yeah, I mean, earlier in the week, I'm sure participants learned about the Unknown Causes of Kidney Disease project that's led by AKF. And through that project, you've been able to identify that 5 to 10% of individuals at end-stage kidney disease don't know the cause of their disease. And so some of that can be attributed to FSGS, and even relevant to just this session, as Dr. Lucas mentioned, some of those end-stage kidney disease, some of those individuals at end-stage kidney disease might have undiagnosed FSGS, and they might have genetic causes like APOL1 kidney disease. And so, you know, there's certain risk factors that might make it more likely for you to be interested or be relevant to receive genetic testing, even if you are at end-stage kidney disease, because that information can both help you better understand the cause of your disease, but it can be really helpful for your family to understand what risk they might have to develop kidney disease as well.
Well, I just want to thank
more about genetic testing at AKF or at nefcure.org.
Thank you, Lauren. Well, I just want to thank all four of you for the work that you're doing in this space. I mean, I initially came into the nephrology field 2003, and seeing where we are now and seeing all the energy is just really amazing. I can't thank all of you for your work, and it's certainly motivating for us here at AKF2 to see two of our clinical scientists and nephrology alumni here as well to present. That's going to wrap our session for today. I want to thank all of our panelists throughout the week. I want to thank all of our audience members for asking great questions and being active in the chat all week long. I can't believe that this is our last session for Kidney Action Week, but that's a wrap. We have some closing remarks coming up from AKF President and CEO LaVarne A. Burton. But you know, you'll see a survey pop up in the chat box. Please fill it out. Let us know how we're doing. And we will see you all next year. And I'll turn it over to LaVarne to close us out.